Okay, so we're going to get started. I'm Sharon Terry, the president and CEO of PXE International. I have to change my name tag on my Zoom since we're using Genetic Alliance's Zoom, which is my other nonprofit, since they have an account that will allow us to have more people than the typical paid Zoom. This is a series of talks that are happening over the whole year. We took the annual meeting and broke it up into discrete talks, which then allow more time for the speaker. And we want to remind you that this is not about you personally. So we are not inviting any personal questions about you or your loved ones and PXE. So nothing about your clinical condition or a symptom you have. This is about PXE in general. And if you have any personal questions, you can email them to me, Sharon@pxe.org, or Mary at pxe.org, and either one of us will get you the answers you need. The other thing I want to remind you is that this is often cutting-edge research. So nothing you see here means that you should run out and take some drug or start some intervention. Li is going to speak to us about therapeutics in PXE. Therapeutic development takes at least 20 years, costs sometimes up to $1 billion, and has a 95% failure rate. So we really need researchers like her, who are very dedicated over many, many years, decades, to do this work. But this is not a talk that gives you information to go and take some drug or to try to find whatever she's speaking about. We will absolutely keep you informed as therapies go through clinical trials and go through the paces they need so that they're useful in humans. Lots of things that we discover in animals don't translate to humans, but they're still absolutely critical steps in the process. So 2 reminders. 1, nothing personal in any of the questions. Please don't talk about your condition. And 2, none of this applies to you today. This is also, everybody has the same background as we do for understanding where we're going as a foundation in terms of trying to find a therapy. So today, as I mentioned, we have Dr. Li, who's going to speak to us about therapeutics and PXE, and she's with the Department of Biochemistry and Molecular Biology at Thomas Jefferson University in Philadelphia. We've worked a very long time with Li. You might remember that she worked extensively with Dr. Qiaoli Li, who passed away last year, year before, and has been dedicated to PXE for a very long time. Jefferson is our one and only center of excellence, and so we're very, very fortunate to have Li and her colleague Kuhn still working on PXE for us with great vigor and enthusiasm, and continuing to make wonderful discoveries that we can then build on. So without further ado, Li, I'll turn it over to you.
Thanks, Sharon, for the kind introduction. So today I will talk about how we research scientists develop therapeutics for PXE. I'm going to start the PowerPoint. I believe everybody in the audience knows PXE is a genetic disease. It is caused by mutations in the ABCC6 gene. Well, the gene responsible for PXE was found 2 decades ago. We and others continue to identify previously unreported mutations in ABCC6 using new sequencing technologies. The ABCC6 gene codes for a protein called ABCC6. The ABCC6 stands for ATP binding cassette. So this is the first 3 letters ABC. And then subfamily C, that's the second letter C. And then 6 is the member 6. So this protein is a transmembrane protein crossing the cell membrane. They are called ABC proteins because they have, and ABC proteins have 2 large intracellular loops that are important for ATP binding and hydrolysis of ATP. So ABCC6 belongs to a family of ABC transporter proteins. There are 48 ABC proteins in human. These proteins have ATP binding domains inside the cells, and these proteins utilize the energy generated from ATP hydrolysis to pump out a wide variety of molecules and compounds outside of the cells. For instance, ABCC1 is a transporter. Because ABCC6 and ABCC1 are closely related, people think that ABCC6 might also be a transporter. However, the molecules transported by ABCC6 has not been identified. The ABCC6 protein is predominantly expressed in the liver, a little bit in the kidneys, but not in the skin, the eyes, and the blood vessels affected by ectopic calcification. It is unclear why, when ABCC6 does not do its job in the liver, causes the distant tissues to calcify. Several studies have established that PXE is a metabolic disease. In healthy people, the ABCC6 protein in the liver serves as a pump and presumably transporting some critical molecules from the liver to the blood circulation. And in the blood circulation, that molecule serves as a powerful anti-calcification factor so that the skin, the eyes, and the blood vessels don't calcify. When ABCC6 does not do its job, so in PXE, the concentration of such molecule in the blood is reduced and causing the skin, the eyes, and the blood vessels to calcify. This molecule was found to be inorganic pyrophosphate. It is a small molecule. It has 2 phosphate groups connected with phosphorus, oxygen, phosphorus bond. And its concentration in the blood of PXEers were reduced by 60 to 70% compared to non-PXE healthy controls. ABCC6 carriers have intermediate pyrophosphate in the blood. So we know pyrophosphate is very important for ectopic calcification. It is present in our urine and plasma samples. The physiological concentration of pyrophosphate is in the macromolar range. The calcium and phosphate concentration, however, are in the millimolar range. So there is a 1,000-fold difference in their concentrations. But the macromolar concentration of pyrophosphate is sufficient to inhibit calcium phosphate deposition in soft connective tissues. Pyrophosphate is important for calcification also because people lacking plasma pyrophosphate suffer from a severe calcification disorder known as generalized arterial calcification of infancy. So this is a very severe vascular calcification disease, and many children die from the disease because of cardiovascular collapse. The cartoon is a summary of our understanding of the molecular pathway from mutations in ABCC6 and a few other proteins to ectopic calcification in soft connective tissues. There are 2 important molecules here, pyrophosphate and phosphate. Phosphate promotes calcification, and pyrophosphate inhibits calcification. The extracellular levels of pyrophosphate is controlled by 2 enzymatic reactions. Pyrophosphate is generated from ATP hydrolysis by a membrane-bound enzyme called ENPP1. Pyrophosphate is cleaved to 2 molecules of phosphate by a membrane-bound enzyme called TNAP, which stands for tissue non-specific alkaline phosphatase. So ENPP1 is critical. So when ENPP1 carry loss of function mutations, that they cause generalized arterial calcification of infancy, because those patients have very, very low pyrophosphate in their plasma samples. So in terms of PXE, the ABCC6 is a transmembrane protein. See here, it crosses the cell membrane. And in the liver, it pumps out some molecules. So at this point, we still don't know the nature of the molecule, but people have found that ABCC6 is expressing cells release lots of ATP, autocytosol cells. And then this ATP serves as a substrate for ENPP1, and then pyrophosphate is generated. So in PXE patients who have mutations in the ABCC6, the extracellular pool of ATP was reduced, and therefore plasma pyrophosphate levels was reduced. So to study PXE in ways that are not possible in man, Dr. Vito's lab at Jefferson developed an ABCC6 knockout mouse model. This mouse develops calcification in the skin, in the eyes, and in the arterial blood vessels. So the calcification phenotype is similar to what we see in PXEers. A unique phenotype in this mouse model is calcification of the dermal sheaths of vibrissae in the muzzle skin. So you see the mouse has the whiskers coming out from the muzzle area. And if we do histopathology of the muzzle skin is different from the skin in other parts of the body. So the muzzle skin here has specialized hair follicles called the vibrissae. So it is a blood-filled connective tissue dermal sheath here that can become calcified over time. We can also monitor calcification in the muzzle skin non-invasively by macro-CT imaging here. So you see the arrows here indicates ectopic calcification, which are consistent with the dermal sheath calcification in histopathology. And this calcification phenotype in the muzzle skin occurs as early as 5 to 6 weeks of age. And the phenotype is progressive. So when the animals get older and they develop more calcification in the hair follicles in the muzzle skin. And we usually use muzzle skin calcification as a reliable and robust biomarker to reflect the progression of the disease. And similar to patients with PXE, the plasma levels of pyrophosphate was also reduced by about 60 to 70% in our ABCC6 knockout mice and also in our ABCC6 knockout rat. So those animals serve as very nice animal model systems to study the human disease. Next, I'm going to show you 3 studies where we tested the effects of treatments in this ABCC6 knockout mouse model of PXE. The first one is atidronate treatment. Because pyrophosphate levels are low in PXE, our initial idea was to administer pyrophosphate to our mice. Pyrophosphate, however, has phosphorus bond, and it is rapidly destroyed by phosphatases in the body. In fact, the administered pyrophosphate is reduced by 50% in just 30 to 45 minutes after administration. We therefore switched our attention to pyrophosphate analogs here called bisphosphonates. Bisphosphonates are stable and they are resistant to biological hydrolysis because they have the phosphorus-carbon phosphorus bond, and that makes the bisphosphonates very stable. And bisphosphonates are used in the clinic for the treatment of osteoporosis and other bone diseases because they have anti-osteoclast activity. And because bisphosphonates share structural similarities with the pyrophosphate, they also possess anti-calcification activities. So here in our study, we choose atidronate. earlier version of bisphosphonates in our mouse. This is our study design. So we took 5 groups for mice, and the first group, so we have 7 ABCC6 wild type mice on the normal diet. So this is our negative control for calcification. The second group is we have 9 ABCC6 knockout mice on the control group. So this is our positive control for calcification. And then the 3 groups are the treatment groups. So we have 6 to 8 mice per group, and all the mice are ABCC6 knockout. And then we administer attidronate in the food. So we added attidronate. So this 1x corresponds to the equivalent dose used for treatment of osteoporosis in patients. And then we increased that dose to 5-fold and 12-fold. And then the treatment was started in the mice when the mice were 4 weeks of age. So this is the time ectopic calcification in the muscle skin has not developed. So this experiment is a calcification prevention study, and the treatment was initiated very early. And then the treatment lasted for 8 weeks. And then at the end of 8-week treatment, the animals become 12 weeks old. And then we euthanize the animals. We collect the muscle skin. And because each mouse has 2 muscles, and we use 1 muscle skin for histopathology and the other muscle skin for calcium assay. So if you look at the left side, so on the upper panel, that is the HE staining. And on the lower panel, that is alizarin red staining. Alizarin red staining is a calcium-specific stain. And here you see 2 colors, some green, bluish color, and then red color. So the red color here represents calcium deposits in the dermal sheets of Fabrice. So you can see here, the wild type mice don't have any calcification. And then if the knockout mice were not treated, we saw loss of calcification in the hair follicles. And if the animals were treated with attidronate at the highest dose level, there was a significantly reduced calcification in the muscle skin. And we could validate the findings by quantifying the amount of calcium in the muscle skin. So we took the muscle skin, we decalcify the calcium crystals in the muscle, and then we used a chemical assay kit to measure the calcium content. So again here, there was significant difference between wild type and knockout mice. And then if the knockout mice were treated with attidronate, this 12-egg attidronate has significant reduction of calcification. So while the results are encouraging, you see that even at high levels, attidronate could not completely prevent the calcification. While we are not the only one who had this observation, other people also found if they target the paraphosphate deficiency by either bisphosphonates or administration of paraphosphate directly, neither of these therapies could completely prevent the calcification. The results suggest that PXE might occur from other paraphosphate independent mechanisms. So, we ask, is there another signaling pathway that is involved in the pathogenesis of PXE? So recently, we identified a pathway taking place not in the liver and the blood, but in the tissues, in the skin, eyes, and blood vessels that are directly affected by PXE. So this pathway is the local activation of DDR/PARP pathway. So the DDR stands for DNA damage response. PARP stands for poly-ADP ribose polymerase. So this was the first paper where this signaling pathway was reported to participate in the calcification process in calcified human arteries. So in this paper, the authors reported that in calcified arteries, the oxidative stress causes DNA damage, and then DNA damage activates PARP. So the PARP are those poly-ADP ribose polymerases, and the PARP enzymes bind DNA, and then the PARP synthesize poly-ADP ribose. So this is PARP. This is the product that is being synthesized by the enzymes. And the PAR is this biopolymer synthesized by PARP. And then that PAR traps calcium in the tissues and forms dense liquid droplets with calcium ions. And the authors also reported that if they inhibit the PARP enzyme activity, and they could block vascular calcification in cultured vascular smooth muscle cells and in an animal model of chronic kidney disease. So we ask ourselves, does this pathway also participate in the calcification process in PXE? So to address this question, we collected the muscle skin from our ABCC6 knockout mice, and we cut slices. We put the slices on glass slides, and we do HE staining. And here you can see those dark purple stained material. That is a calcification. So this is a knockout. This is wild type. And then we stain the minerals using a calcium binding fluorescent dye. So this fluorescent dye stains the calcium deposits as a red fluorescent color here. So that is where calcification is in the muscle skin. And then on the same tissue section, we stain the section with an antibody against H2AX, which is a DNA damage response marker. So this gives us a green fluorescent signal here. And then if we overlay the DNA damage response marker and the calcification, and we saw a very nice co-localization. So this happens not only to DNA damage response marker, but also for the end product of the pathway, the PAR. The PAR also co-localize with calcium dependent. So these results suggest that the DDR PARP pathway is also involved in the calcification process in our PXE mice. So now we ask ourselves, if we inhibit this DDR PARP pathway, can we also block calcification in the mice? So we designed two treatment studies. The first study was a genetic approach, and the second one was a pharmacologic approach. So in the genetic approach, we crossed our ABCC6 knockout mice with a PARP1 knockout mice. And then we generated the double mutants. So here, this is the wild type. This is just ABCC6 knockout wild type for PARP1. And those mice were knockout for both ABCC6 and PARP1. So the PARP1 protein was not expressed here. So, and then if we look at the calcification in the muscle skin, so here, this is a histology. We saw reduced calcification in the double mutants. And then if we quantify the calcium content in the mice, so the panel B shows statistically reduced calcification in the double mutant compared with the ABCC6 knockout. So now the genetic approach tells us, inhibition of the pathway inhibits calcification. So is there a DDR pathway inhibitor that we could test? And the answer is yes. So minocycline, so antibiotics that has been used in the clinic for many years, inhibits the DDR PARP signaling pathway. So in this study, we administer minocycline in the drinking water for our ABCC6 knockout mice. And then at the end of the study, we perform histopathology. And again, we saw reduced calcification in the mice treated with minocycline. And then if we quantify the calcium content in those mice, there was a significant reduction in the mice treated with minocycline compared with the non-treated control mice. So far the results are encouraging. So does this pathway participate in calcification in PXE patients? So again, we repeated the H2AX and PAR immunofluorescent staining from skin tissues from PXEers. Here we have 3 to 4 PXEers lesional skin biopsies, and we have 3 to 4 control samples from healthy control skin biopsies. And then on the left, so that was Fancasa staining, which is another calcium-specific stain. And you see those dark black materials, the calcium deposits, and that was negative in the control. And again, so we stained the minerals as red fluorescence, and we stained the DNA damage response marker and the PAR in green fluorescence. And if we overlay the green and red fluorescence, we saw a very nice correlation, a co-localization between the two signals. The results suggest that in addition to the mice, this pathway is also involved in PXE patients' skin. So to ask if inhibiting this pathway could be helpful to inhibit calcification, we cultured the fibroblasts. So they are the dermal fibroblast cultures from healthy control people and from PXEers. And if we culture those fibroblasts don't calcify themselves, but they calcify if we added phosphate in the cell culture media. The phosphate induces the cells to calcify. And then if we added minocycline in the calcifying medium, and we saw a robust difference in the amount of calcification. So here, the phosphate induces the cells to calcify, and the minocycline treatment significantly reduces calcification, and the quantification was shown in the panel B here. So the minocycline also inhibits dermal fibroblast calcification in PXE patients. And the signaling pathway was also reported in Oliver Wanneker lab in Belgium. So our studies were independently validated by another lab. So far, we identified 2 possible mechanisms for PXE, the systemic and the local factors contributing to ectopic calcification in PXE. The metabolic nature of PXE is supported by reduced circulating concentrations of paraphosphate, and attidronate prevents calcification. And on the other hand, the local activation of the DDR-PARP pathway triggers calcium deposition in tissues susceptible for calcification. And here, by intervening this pathway, minocycline also prevented ectopic calcification. So remember, neither attidronate or minocycline could completely prevent calcification. So now we ask ourselves, can we reduce calcification further if we target the 2 pathways at the same time? So here we designed a 3rd experiment. So in this experiment, again, we have the wild type mice and the knockout mice. They are not treated. They serve as negative and positive controls for ectopic calcification. And then this group were a group of ABCC6 knockout mice treated with attidronate. And then we have another group of ABCC6 knockout mice treated with minocycline in the drinking water. The last group consists of ABCC6 knockout mice receiving both attidronate and minocycline treatment, because we want to answer the question whether a combined treatment would do a better job than either treatment alone. Again, this experiment was a calcification prevention study. The treatment was initiated in the mice at 4 weeks of age before calcification takes place. And then this is an 18-week long treatment. And then at the end of the treatment, we euthanize animals. And this time we used a macro CT imaging technique to quantify the amount of calcification in the muscle skin. Here, the wild type mice don't calcify at all. And then the knockout controls calcify a lot. And then if those knockout mice were treated with etidronate, and we saw less calcification, and also less calcification for minocycline treatment. And we saw much less calcification if the mice received both etidronate and the minocycline treatment. And then the results were quantified by measuring the calcification volume from the reconstructed macro CT images. I hope you agree with me that the calcification was less in this combined treatment group. We also analyzed the calcification in the second piece of muscle skin using histology and calcium assay. So this is Fancasa staining. So the calcification was stained as dark black material. Calcification was negative in the wild type, and there were lots of calcification here in the knockout, and there was less in the etidronate and the minocycline treated mice, and a lot less in the combined treatment. And the results were confirmed by measuring the total amount of calcium in the samples. So that comes to a little summary of my talk. So a detailed understanding of disease mechanisms enables us to design rational therapies in our mouse model. Etidronate treatment and minocycline treatment alone prevented ectopic calcification in a ABCC6 knockout mouse model of PXE. And then the combination of etidronate and minocycline is more effective than either treatment alone. So the results are very encouraging. As Sharon talked to everybody at the very beginning, so the message here is not for you to quickly jump on any of those treatments, because the studies were performed in mice. And the mice are not simple and small humans, and the humans do not have whiskers. And our mice are inbred, so they are genetically the same. Our animals have the same genetic makeup. Well, we are not. Our mice were housed under a controlled environment. They have the same light, dark cycle. They receive the same food and water. They are all pathogen-free. Our animal studies are proof of concept studies. So I tell people the animal results give us the proof of concept that the treatment could be helpful, but very often animal studies don't accurately mimic how the human body and human diseases respond to drugs, chemicals, or treatments. And the results derived in animal studies may not work in humans. Therefore, carefully controlled clinical trials are needed to confirm or dispute the clinical benefits of therapies. And clinical trials are also needed to evaluate long-term treatment effects. So before I finish, I would like to thank everybody in the lab who contributed to the project. We would like to thank PXE International. PXE International has supported our research for many, many years. I would also like to thank the PXEers who have contributed to many of our research projects. Thank you very much.
Thank you, Lili, for an excellent talk and for all your work. You can see that Lili has done an enormous amount of work. The floor is open for questions. You can put your question in the chat. You can raise your hand either physically or with a hand waving motion, or you can unmute if you can't find the controls. Okay, so are further studies planned? What further studies are planned, Lili?
So we are now exploring new therapies. So at the moment, we are looking at the effects of paraphosphate. So we did not consider paraphosphate in the first place because, people know that the paraphosphate will be cleaved very quickly in the body. But now we are considering paraphosphate. We are administering paraphosphate to our animals and see if there's any effect. And also, we would like to develop a one-time fusion, one-time treatment approach. We are developing gene therapies for PXE. So if we already made AAV-based ABCC6 gene vector, and we hope that this therapy would be just a one-time infusion, like one-time injection, that will result in long-lasting protein expression in the liver. Of course, everything will be done in our animal systems.
Great, thank you. Alex's hand is up, and then I have a couple questions in the chat. Alex?
Thank you, Sharon, and thank you, Doctor, for a really, really excellent talk. I admit my training is very far from this, so I'm sorry if my question is something that you covered and I just didn't understand. But when it comes to the results that you were sharing about the minocycline and related therapies, is that only for preventing calcification or minimizing calcification that may have already taken place?
That is a very good question. So our studies were designed to prevent. So they are prevention studies. So our mice calcify as early as 5 to 6 weeks of age, but we administer the drugs, before calcification takes place. So this is the calcification prevention study. We also performed the same treatment in older animals where they have already developed calcification in tissues. So what we found was that the treatment could prevent new calcification from happening. So it stops the progression of the disease, but does not remove existing lesions.
Thank you so much. And a quick follow-up question after that clarification. Is there any data on, or does your study show anything in regards to like the length of this treatment protocol and or what happens when it stops afterwards? Were there follow-up studies or continued samples taken from mice who had received the treatment for X amount of time and then stopped?
Yeah, this is a very nice question. So in terms of the length of the treatment, so because the calcification is progressive, and as I said, many of our studies are just proof of concept studies. So we usually perform a short-term treatment study. So in the third project I presented, that was 18-week long, and I thought that was really long, that we have to wait for 18 weeks. If we could ideally, if we wait for 1 week and we saw the results, that would be nice. But so we wait until the knockout mice develop a robust calcification phenotype. That is normally like 2 to 3 months of time. And then we stop the study. So we have not really looked at, let's discontinue the study and see, will calcification come back? We have not looked at that, but my assumption is that calcification will come back.
I see. Thank you so much. And the last thing I'll say, I'm sorry, I want to be mindful of everyone's time, but would it be possible, or is this better to go through you, Sharon, if we have further questions or without disclosing anything personal, insights we might share that relate specifically to what you've shared today, would I be able to email to get in touch and share some information?
Yes, so if you email me, I'll connect you to Li.
Thank you so much. This was wonderful. Thanks again.
Great, thanks for your questions, Alex. And Colleen says thank you for the sessions, and they're great for people who live with PXE, which is everybody here pretty much. And then Anne, I think Li just answered the same question Alex asked, which is, can it cause regression. And, we'll say the same thing we've been saying almost every webinar is nothing is known yet that will reverse calcification, particularly in organs that turn over very slowly. So, things like the liver can turn over more quickly and you can donate half your liver even, or other organs have a regeneration time that's more quick. Skin does not, retinas don't. And so, yeah, we still don't have anything that will address that directly. We do have one scientist that you've heard from, John Bonacker not Bonacker, oh gosh, my brain just went. Anyway, a guy at Vanderbilt that we're supporting who is looking at macrophages essentially eating, that's a really terrible lay term, the calcification to see if we can get regression that way, but we haven't been able to figure that out. Calcification is a massive field. And so anything that's discovered in this works also, hopefully, in things like scleroderma, which is another rare disease, or common conditions like hardening of the arteries is the kind of usual name. Other questions? Okay, so I'll just say, Alex.
I'm so sorry. I have one last question. I'm curious if there's any insight to share about how far we might be from human clinical trials into the specific research that was shared today, now that sort of proof of concept has brought some promising results. Is there any insight into time frame, whether that is a year or 15 years, or is it still too early for that?
So I will first start with the etidronate. Dr. Wilco Spearing in the Netherlands has run etidronate trial in patients living with PXE, and he published several papers already, and his results already demonstrated that etidronate improves calcification. I think he's running more trials now. If you go to clinicaltrial.org, you can find all the registered clinical trials related with PXE. Sharon, I don't know whether Wilco will give a seminar
Yes, he will.
on his etidronate clinical trial.
Yeah, and he also, we've already recorded a couple from him. The problem with his trials have been that the regulatory agencies like FDA and EMA in Europe are not accepting the difference he's seeing is not statistically enough for them to think that this is a good treatment, which is why Li studies with the minocycline are really excellent, because maybe the combination is better, clearly better. And also the problem with etidronate is it was taken off the market because it causes necrosis of the jaw. So it kills the cells in the jaw if it's used for a long time. It was typically used for people who had osteoporosis in their 60s and 70s, and maybe they took it 10 to 20 years and then they died. With people with PXE, we'd want to be giving this somewhere around 18 or 19 years old once bone development has slowed down. And there's no way that the safety profile is good enough for us to do that. I will put his name in the chat, and you can find his talk on our YouTube channel.
Yeah, and also his clinical trial is a 1 year long trial.
Yeah.
So maybe that is another thing to consider.
Right. Yeah, and this is back to, I'm a broken record on this. Those of you who have come to these webinars, the trials are going to have to be very long to show anything in PXE, because the people who are in a trial 1 year could have maybe not had any progression for a year anyway. So we can't say it was the drug that changed it. The mice are great because they age quickly, and we can watch the phenotype of the whisker, the bristle, change. So that's fabulous. The other thing is we have no biomarker, and you've heard me say that over and over and over and over and over, which is the FDA and EMA so far won't accept anything that we offer in terms of what's the way we're going to measure whether or not the drug is working, because it's so slow, because it's so variable, because one person, even compared to their sibling, has a different course of disease. And then to answer more generally, so if Li comes upon the perfect combination in mice here, Then our next task is to find someone who will sponsor a human clinical trial, because Lili's not going to do that. And the Jefferson Center of Excellence can't do that because, again, I did say at the beginning, it could cost up to $1 billion to take a drug through. It's at least going to cost many, many tens of millions of dollars. And so we need a company. We need a company to get interested. We need a company to believe and be able to tell their shareholders, stockholders, that they will, in fact, make money on this, which is hard to do in a rare disease because, you know, we know of 5,000 of you worldwide. Not everybody would even need a drug because not lots of people don't have severe symptoms at all. And so we're probably looking at, you know, if Lili tells us this year, this is it, this is the best thing, then we will shop around for a company. It would probably take 2 or 3 years to set up the phase 1 clinical trial to make sure that this is not toxic to give to humans in this combination. Phase 2 clinical trial to look at, can we escalate the dosing? That's where we are with Daiichi Sankyo, and the TNAP inhibitor trials just finished the phase 2. And then go to a phase 3 trial where lots of people would take it and we would map them over 4 or 5 years to see if whatever we pick as a biomarker is actually changing. And so I would say if Lili makes this amazing discovery this year, we're looking at a minimum 10 years before we would see an approved drug from it.
Hopefully. Yeah.
All right. And Mary put in the chat clinicaltrials.gov. You can look up the clinical trials there. Note that lots of them are no longer active trials. You can actually click a button that says active and not active and so on. Okay, anything else? Lots of thank yous in the chat. Thank you, Lili, for your work. I'll also say that you'll be hearing from PXE International. Everyone will be. Because what we do is we keep Lili and her work supported. So we raise money that then Lili and her colleague Koen Van Bettering at Jefferson both do this incredible research that's absolutely critical to our getting the job done. And also have done some even clinical things, like Lili came to Washington, D.C. and took the blood of people with PXE to see if their pyrophosphate levels were low. And that was cutting edge because we didn't know for sure if they would be low. And so we learned over the course of a day how PPI changes during that day and how the range of PPI levels in people with PXE. And we're about to do a small study looking at, are all labs measuring PPI the same way? We know the answer is no. And so we're going to try to figure out what is the best way to measure PPI, because not even that is done in any kind of standard way yet.
Yeah, thank you, Sharon, for the invitation. Thank you all. Have a nice day.
Yes, you too. Thanks, everyone.